you have any questions? The new material we're going to be starting on after we finish up a little bit of material structure today, it's a little confusing. Has anybody tried the website again recently?
It doesn't.
How are you trying to get to it? Are you going to Franklin College and academics? And
My website.
Do you get it?
I
am.
tried doing that. I tried to use it too.
so I tried it this morning and it worked on my computer.
If you press on the link immediately, it won't go. You have to go through Franklin College.
You have to go through Franklin College Study.edu.
Or you have to go through Franklin College.
I will try.
So go to Franklin College, academics, majors, biology, that gets you to the natural science. And then it's course info, I think, is the button. And then it takes you to the list of courses, yeah. But anyway, the next topic we're going to talk about is bacterial classification, Bernie's manual, taxonomy. So the lecture notes are titled Classification 2012. That's what's in Dropbox. Okay. The PowerPoint is titled Bernie's Overview 2014. So if you're unsure of which things corresponded to the next material, that's what it is. I apologize for the trouble they're causing us with these website things. Technology. Okay? So we were in the process of talking about structures you might find in the cytoplasm of some bacterial species. And we're talking about inclusions. Remember that inclusions contain typically storage materials that cells will accumulate that they can then draw on during bad times. Or sometimes they're the product of metabolism, like the anoxygenic photosynthesis we mentioned at the end of the class. This is a micrograph of PHB granules. This is one of the stains that you will do in lab in a couple of weeks. This particular granule, there's several ways you can do the staining process, but this was stained with a red fat-soluble dye called Sudan-3, and Sudan-3 preferentially binds to those PHB inclusions. I don't know if you remember Sudan-3 from the cell class. We had the biological molecules lab, you may remember, where we did tests for protein. We did the Bio-Red test, we did the Benedict's test. Does anyone remember how we did fat stain? Little pieces of filter paper. You put the little droplets on there of various foods and controls, and then you put them in a finger bowl, which is probably sitting over here, that red stain. That was the Sudan-3 stain, and that preferentially found the fats, and then you wash them off with water. It retained the fat stain and sent down to the fat granules. So this is a similar kind of staining process right here with the Sudan-3. I mentioned sulfur granules. Another type of granule that is not atypically stored in bacterial cells are long polymers of phosphate groups. They're called polyphosphate granules. What do you need phosphorus for in metabolism? What kind of molecules can we make if you have phosphorus mutation? ATP? Nucleotides? Okay. So this is an important nutrient to have available, and some microorganisms can store this in their cytoplasm. These polyphosphate granules in particular have this ability when certain dyes are exposed to the organism that the granules change shape and change appearance, not shape but appearance. That change in appearance and color particularly is called metachromacy, and sometimes these particular granules of polyphosphate are called metachromatic granules because of the weird kind of visual appearance they give when they're stained with the various stains. In particular, there's a stain that gives you really marked metachromacy called the volutin stain. And so sometimes these granules are also called volutin granules. This is another one that's kind of tricky, and this is one we used to do along with flagella stain. But again, the results were so spotty and so few people got good results that we stopped doing this particular one. But if you want to look this up online, Google it sometime, look at metachromatic granules or volumes of granules, you can see what that particular appearance looks like. Sometimes different species of microorganisms store interesting things in granules, like iron. Here you can see the cytoplasm of this particular cell. It's a little bit hard to see here. Has all of these iron granules lined up on this particular edge of the cytoplasm of the cell. These particular iron granules form a structure called a magnetosome, and magnetosomes allow these species of bacteria to orient themselves into magnetic fields. What's the purpose of that? No, not really sure. There's a lot of theories about what magnetosomes might do, but they think that they might somehow allow bacteria to orient in situations where they get favorable exposure to oxygen or to various nutrients, but we don't really know absolutely for sure. But again, that is a species-specific characteristic of some types of bacteria. Okay, remember that to be a cell, you got to have ribosomes. But also remember that ribosomes are different in prokaryotic cells than they are in eukaryotic cells. And that difference is oftentimes expressed in terms of the S value or Svedberg constant. Eukaryotic cytoplasmic ribosomes, what's the S value of those? 80S, okay? Prokaryotic ribosomes, S value 70S. The ribosomes, the 70S ribosomes, like the eukaryotic ones, consist of two subunits. In this particular case, the large subunit is the 50S, and the small subunit is the 30S. And again, those values are not additive. 50 and 30 does not equal 70. Okay? But the importance of understanding the fundamental difference between prokaryotic and eukaryotic ribosomes is that certain drugs, certain types of antibiotics, can selectively bind to 70S ribosomes and therefore interfere with protein synthesis. And obviously, if you mess up protein synthesis, that can be lethal to the cells. Are 70S protein synthesis inhibiting antibiotics completely selective for killing bacterial cells and not eukaryotic cells? They're not. Why not? Mitochondria have 70S ribosomes. Okay? If you're only on a 7 to 10 day course of antibiotic, that inhibition of mitochondrial protein synthesis is not of really any consequence, and therefore there won't be any toxicity to the cells. But for example, if you are being treated for tuberculosis, you can be on a year or longer regimen of antibiotic, and therefore there's got to be some very careful monitoring of your cells in your blood to make sure that the inhibition of mitochondrial protein synthesis doesn't destroy particularly bone marrow cells. If you start destroying bone marrow cells, then you get decreased immunity, decreased red blood cell content, etc. So that's pretty serious stuff. Ribosomes are usually found in the cytoplasm of the bacteria as free ribosomes, but occasionally they will be found also on mesosomes, and many times they're also attached to the cell membrane itself, allowing the bacteria to do some secretion. Because remember, you can't secrete proteins unless the ribosomes are associated with membranes. You also have to have DNA in order to be a cell, but the DNA does not have to be enclosed in a membrane-bound nucleus, and it is not in a prokaryotic cell. So consequently, we don't use the term nucleus when we're talking about prokaryotes, and instead we refer to the nucleoid region. Remember, the bacteria have a haploid genome. They typically have a single circular chromosome, just one. Although sometimes there will be small pieces of extra chromosomal DNA in addition to the main chromosome. Those would be the plasmids. Remember we said that the plasmids can potentially be transferred from one cell to another by conjugation through those sex pili. The DNA that makes up this nucleoid region is highly, highly supercoiled. And then there's an enzyme that is involved in this supercoiling process, and particularly that allows the coil to form and then to relax again. It's very important for that coiling to be relieved and relaxed during DNA replication. So there are enzymes that control that. There are a class of antimicrobial drugs called quinolins, which can inhibit these enzymes that control that state of DNA between a supercoiled and a non-supercoiled state. And these particular antimicrobial drugs are pretty effective at killing bacteria because they interfere with this enzyme. Some of you may have taken this particular class of antibiotics. Cipro belongs to this class of quinolin antibiotics. Cipro, I know, is commonly used for treating traveler's diarrhea, for example. It is also the antibiotic of choice for anthrax. So years ago when we had the original 9/11, soon following that was the anthrax scare, where anthrax spores were mailed to members of Congress. And we started to worry about the supplies that were available of Cipro if we were to have a big anthrax bioterrorism threat, and whether there was enough of that antibiotic on stock to treat the people that might be affected. Bacterial genome approximately has about 3,500 genes, so relatively small genome. At any one given time, only about 1,000 of those are probably being expressed at any given time. I mean, some genes have to be expressed always. I mean, think about it. Genes for the enzymes that are involved in glycolysis. Cells always have to have glycolysis, okay? Regardless of whether they ferment, regardless of whether they're doing aerobic respiration, anaerobic respiration. You always need those particular enzymes. Genes that code for proteins that are always being expressed, that really are never turned off, are called constitutive genes. Okay? So some of this 1,000 are constitutive genes. But bacteria have a remarkable ability to respond to changes in their external environment. Why express the gene, for example, for beta-galactosidase, which is the enzyme that breaks down the sugar lactose, if that substrate isn't present? And so bacteria have a control mechanism that allows cells to differentially turn on and turn off genes that allow them to respond then to changes in their external environment. What are those control elements called? Remember? You had both cell and you had genetics. Operons. Okay? So we have a lac operon, we have a trip operon, and those particular regulatory elements, remember the operator, the repressor proteins, allow you to selectively turn on and turn off genes as needed. This is kind of a cool picture because this shows a bacterial cell that has been lysed and the DNA has been released from the cell. It is also uncoiled, so it's not so supercoiled as it normally was. But this gives you an idea of how supercoiled the DNA has to be for it to normally fit into this little region kind of in the center of the cell, that nucleoid region. And then finally, also in cell biology, we talked about the eukaryotic cytoskeleton and all of the different kinds of protein elements that make up the cytoskeleton. I don't know if you remember that there were microfilaments, microtubules, and actin filaments. They form this meshwork inside the cell that has all these different functions. Cytoskeleton is involved in mitosis, meiosis, segregation of chromosomes. It is involved in maintaining cell shape, cell structure, and constantly is assembling and disassembling. It's a very dynamic structure. Well, we used to think that prokaryotic cells had any kind of cytoskeleton. But some of the more advanced, particularly fluorescent antibody staining techniques, have now indicated that there is a very simplistic and kind of primordial cytoskeleton in most bacterial cells as well. You can see it here in this particular slide. There are particularly actin filaments that permeate through the cytoplasm and play some roles in maintaining cell structure and cell organization. Okay, so that's kind of a brief overview of some of the cell structures you might be expected to see in some cells. And again, this basic knowledge of the cell structures is going to come back and be important in lab as we start learning various staining techniques to allow you to identify these particular structures. And that's going to provide us with some important morphological information about your unknown. And understanding what the morphological characteristics of your unknown is one of the very first steps in being able to identify that particular microorganism. So the first part of the course, up until the first lab exam, is going to really depend upon a lot learning about physical characteristics of your unknown by doing these various staining techniques. Do the endospore stain, do a gram stain, do the PHP stain, do a motility test, etc. And then we will gather some information that will allow you to start narrowing down what particular type of organism your unknown might actually be. But that's not enough to really do the identification. And so the second part of the course is primarily going to be involved in biochemical characterization of your unknown. And particularly trying to see what happens to these bacteria when we feed them certain foods. Number one, can they metabolize them? Do they have an enzyme, for example, to break down DNA? And then once they have the nucleotides, they can potentially metabolize those nucleotides for some purpose. The other thing we like to see is if they do have the enzymes to break down the big organic molecules into the smaller monomers, what do they produce as a byproduct? And that's particularly going to be important when we talk about bacterial fermentation. So we're going to feed your bacteria glucose. Number one, can it utilize it as a food source? Number two, if it can, what does it produce? Does it produce acids? Does it produce alcohols? And that will give us some additional information about the enzymes that those particular bacteria possess. And what determines what enzymes you have? Genes, right? Well, that leads us into the next topic. And I think this will be the breaking point after we finish this material for the first exam, which is coming up not next Monday, but a week from Monday. Okay? 28th, I believe. If you haven't been working on your study guides, I would really strongly recommend that you do that. Okay? You can now do the introductory study guide. You can do the cell structure study guide. And like cell biology, questions that appear on the exam are oftentimes, sometimes identical to study guide questions, if not variations of those. And so the study guides really help you to organize your studying. They are typically written in such a way that they ask questions in the order in which the material is presented in the lecture notes. So if you have those lecture notes, you can go through and start answering study guide questions and really get practice kind of writing out answers to those particular questions. So keep up. So we're going to spend some time talking about bacterial taxonomy and bacterial identification, which is a good lead into Lab Wednesday. Because on Wednesday in lab, you'll learn to do an important fundamental technique, and that is the streak flicking. And that will allow you to isolate bacteria from a mixed culture and isolate peer cultures from that mixed culture. And you're going to be streak plating all semester long. Okay? Very important technique. The other thing is you're going to get your unknown. Okay? And I'm going to spend some time going through the unknown with you, talk about how you need to maintain it, how often you should subculture it. We'll talk a little bit about the actual process of identifying your microorganism, introduce you to a couple of keys you should be aware of, and kind of set the stage because you're going to be working on this unknown for the rest of the semester. So this is good timing in terms of talking about this particular topic. Well, one of the questions we need to address kind of immediately is, why do we need a classification system for bacteria? Why do we develop classification systems for organisms? Hudson? Part of it is being able to identify them, okay? And part of identification is assigning a name to the organism, right? You know what that process of assigning a name to an organism is called? It's called nomenclature, okay? But we can't start sorting organisms into groups unless we have names for them. Well, when we talk about nomenclature, we need to distinguish between common names and scientific names. So, for example, out on campus, you might see some sugar maples. Is that a common name or a scientific name? That is a common name. Okay. Scientific name for sugar maple is Acer saccharinea. What's the difference between common names and scientific names? Scientific names are Latin. And a scientific name always has two parts. What are they? Genus and species name. Okay? Genus name always being capitalized, species name lowercase. How should you write scientific names? Well, if you can write it in italics, you can do it as italicized. What's the alternative? Underline. Okay? Scientific name should be underlined. What's the problem with common names? What's the problem with sugar? Awesome.
Some regions like false.
Yep. The same organism might have two different common names in different regions, for example. And so scientific nomenclature is a way to standardize and apply one name only no matter where you are. Who is considered to be the father of modern binomial nomenclature? Good. Linnaeus. Remember your botany a little bit. He was a 16th century, 1700s botanist, and he developed this system of nomenclature and classification. And Linnaean taxonomy, assigning things to group, was broken down into these various groups that become increasingly more and more inclusive. What does the K stand for? Kingdom, family, class, order. Whoops, phylum. Class, order, family, genus, species. In plants, what is the equivalent of phylum? Botany? They don't talk about phylum in botany. Bryophyta, Coniferophyta, their divisions. This has become the basis of the old five-kingdom approach to classification. But now that we are in the new world of taxonomy, we have added a higher level of organization, the domain. And the domain now is the currently accepted method of classification. This is the most broad category. We have the three-domain system of classification. Now, these identification and classification systems suffer from a couple of sometimes competing roles. They have a practical role, and they kind of have an academic role as well. The practical role of these systems is identification, figuring out what the organism is. The more kind of academic role is to demonstrate phylogeny or phylogenetic relationships. What does phylogeny refer to? Yes. Phylogeny deals with evolutionary ancestry. Okay? So kind of inherent in this classification system of kingdoms and domains is this idea that if you are on the same family, you are more closely evolutionary related than organisms that are in a different family. And as you move down this particular classification hierarchy, the farther down you get towards species, the more closely related organisms are in individual groups. So this idea of evolutionary relatedness is kind of inherent in this academic role of taxonomic systems. But sometimes these two roles kind of conflict with one another and are a little bit at counter purposes, and that's particularly true in the prokaryotic world. And I'll explain why that's the case in a minute. One of the reasons that classification and taxonomic systems were developed in the first place was to deal with complexity. One of the natural things that the human mind likes to do is, if they're faced with all kinds of complexity, start putting things into groupings to keep better track of them, to organize them. Okay? Well, is there a need to organize the prokaryotic world? Is there a lot of diversity in the prokaryotic world? And the answer is yes, and much more so than we'd ever thought in the past. This is one of the 4 volumes of Berge's Manual. And we'll talk about Berge's Manual pretty extensively in the next couple of days. This was done in 1984, and basically this represents the known information about prokaryotes at that particular time. And so 4 volumes worth kind of indicates the totality of organisms that we were aware of in 1984. And there were about 5,000 species included in this edition of Berge's Manual, about 1,000 different genera. And even that amount of complexity is kind of overwhelming to the mind. And so organizing these things into groupings made sense. It was a way to deal with that particular complexity. Well, as it turns out, this is just the tip of the iceberg. Okay? One of the reasons why we have in the past underestimated the number of different types of microbes out there in the world is that to identify them, the first thing we have to be able to do is grow them in the lab. But many microorganisms that are out there in the world cannot effectively be grown in the lab. So for example, we used to think that seawater had very, very poor microbial diversity. Now we know that that's not the truth at all. One of the ways we've gotten a better handle on microbial diversity in the world is doing some genetic screening, taking samples and finding unique sequences of DNA or RNA. And then each of those unique sequences, we believe, represents different species of microorganisms. So we infer their existence even though we can't culture them and grow them in the lab. And so these new genetic techniques have really allowed us to expand our understanding of how many different microorganisms are out there. And that knowledge has just magnified this need to come up with a classification system to deal with all of that particular complexity. Let's take a look at a typical practical identification scheme for a second. This one's hard to see. Let me flip up the light for a second so you can get a little bit better look at it. Zoom in a little bit. Okay, this particular practical identification scheme is the kind of thing that might be used in medical diagnosis to determine what particular type of organism might be causing a particular type of disease. And they've got this particular scheme broken up into three major categories. Bacteria with gram-positive cell walls, bacteria with gram-negative cell walls, and bacteria with no cell walls. That's down here at the bottom. Okay? So cell wall structure is pretty important in this classification system. And then let's just take a look at the gram-positive bacteria, for example. Cocci in chains, cocci in pairs. Okay? So morphology of the cells is an important criteria. Down here, again, in the gram-negative bacteria, cocci, rods. Okay? Going back up here again, aerobic. Down here, aerobic. There will be situations where we have facultatively anaerobic or anaerobic. So a little bit of physiology creeping in here now into this organization system. Spores or not spores. Again, mainly structural information. So practical systems tend to rely a lot on morphology and maybe a little physiology. But even just using those criteria, you can begin to develop schemes that allow you to fairly easily identify major types of bacteria. Remember that I said that one of the easiest ways to diagnose the TB bacteria is to do the sputum sample and to do an acid-fast stain, because that particular genus is one of the few that is acid-fast. So basically on just structural information alone. So these particular practical systems are really designed to give rapid identification of the organisms. Things are not organized with any intent to try to show evolutionary relationships within those groups. And so these particular classification schemes are not phylogenetic at all. They're said to be phonetic instead. So these practical identification schemes are phonetic schemes. They don't attempt to show phylogenetic relationships. Ultimately, The greatest indicator of phylogeny is how many genes do you share? And what genes do you share? That's the ultimate acid test of being evolutionarily related. Well, remember, genes code for proteins, particularly enzymes. And enzymes can both determine biochemical characteristics, like whether you can metabolize a certain food, but enzymes also can determine, as will other proteins, structural features of the cell as well. They also determine the physiology of an organism. So when we're looking at things like structure and physiology, we're not looking directly at the genes. We're looking at the genes once removed. We're looking at enzymes, which are a reflection of genes. And that's one of the reasons why these phonetic classification systems are very poor at illustrating phylogeny as well, because you're just looking at things too far away. The new phylogenetic schemes, like the 3 domains, are based primarily on genetic information. And they're a much better indicator there